current technologies available. What lab-on-a-chip people have done is develop a lab-on-a-chip device in PDMS, the squishy material that we've looked at before, where we've got our 4 chambers and very small microchannels. And you can load cells into the cell body chamber, and exons are kind of curious by nature. So they start finding themselves away, and they like finding themselves away through a very narrow hole. So what happens if you see this, the neurons into the chamber here? The curious exons actually go through this very small channel, which is 5 by 20 micron. Really, really narrow gap. Way too small for these cell bodies to get through. But the exons themselves, they can find themselves away through here to the exon chamber. This is an idea that was published in 2005. And in 2019, more than 100,000 devices were sold. So very simple device, very popular amongst neuroscientists. The way that the device looks like is that we've got the somal side, the side where the cell sits, and we've got the axonal side where the exons go to. We've got a little bit of a step here because the neurons kind of like having a roof over their head. And then here we can see the small microchannels that's there for the exons. So we've got the reservoirs, they're 3 millimeter high, so that we've got enough place to put the PBS, the buffer in. We then have 100 micron home for the neurons to feel happy and confined in. And then we've got a 3 micron high, I thought it was 5, but it's 3 micron high channel that is connecting the somal and the axonal sides. So if we load our cells into the somal sides, then we get our dendrites being developed in the chamber so that the cells can communicate between one another. And we get the exons being formed all the way to the axonal side, which allows us to do some potential drug treatments on the exons only without disturbing the soma or vice versa. So that's an example of having a selective drug treatment, because this is a very long distance to diffuse. You can buy them with 250, 450 and 700 micron long channels. So this drug won't be able to diffuse over a reasonable amount of time to the somal side. So we can selectively treat the exons in this system. When we started this project, our aims were that we wanted to design and fabricate an in vitro model to isolate an axonal stretch injury using one of these PDMS devices. And we had an idea how to do that. And then look at the axonal alterations following the stress injury and test some potential therapeutic agents. So this was a project we've done together with our Medical Research Institute. We've talked about valve and pneumatic valves and the quake valve yesterday, which was pretty much the small balloon used to potentially make a peristaltic valve. So if you apply air pressure to a thin PDMS membrane, it kind of inflates like a little balloon. And we've seen that for valving and pumping earlier. But our plan was to actually use that under an axon to locally apply a stretch injury. So our plan was to take the quake valve that was normally used for fluidic control, but combine it with a neuroscience microfluidic device so that we could exactly know which axons we were stretching. And we could also very precisely know how much we were stretching them, because we could control the curvature of the balloon by controlling the pressure that we apply to the device. So luckily, the people that first published the quake valve actually put on the website of the research group, they put together a complete list of building the package yourself. So what you need is a range of, in their case, they wanted to operate a whole load of different valves. So you need some valves to open and close to apply the pressures. You need some blunt syringe needles to connect to your fluidic device. You need the device to measure the pressure. And you need some electronics to control all the valves there, all the solenoid valves to get the air to where you want to have it. But luckily, all of this was on a shopping list, so you can just click and order the relays in. There was a simple LabVIEW program that you could also download. And if you brought the same components and put it onto a board, that program actually allowed you to directly control the pressure at the different outlets. So you didn't need a real background in electrical engineering, but no need, you shouldn't be too afraid of wires either, because this was a setup that you got the parts in and you have to all assemble it yourself on the benchtop. And the other thing that we did was using an optical profiler to actually see how much stretch we could create. So we calibrated the amount of deflection of the membrane we could measure for different pressures being applied. So here pressures going from 0 to 40 psi, and we could measure the amount of microns that this balloon actually went up. So here you can see the optical profiler looks at how light bounces back, and if it bounces back faster, it knows that there's an object in the way. So we know how our inflated balloon looked like, and what our experiments were going to be is looking at different pressures when we've got an axon sitting here across, stretching it to how far we could inflate the balloons. So this is the way that the device looked before we were going to use it. So here we've got our grooves where the axons are in, and here we've got our channel sitting underneath our little ballooning channel to apply the axon injury. And indeed, after we'd injured the axons, we could see that there were some blobs appearing here, and that's called axonal beading, and that's the first biochemical indication that your axon is actually really feeling unwell. Here you can see some more disconnectivity happening. And after an hour, you can see that the whole thing shriveled up. So our device was effective, and sadly, we were killing neurons with it. I don't think we need to go through what else has been done in the literature. So we've got our little axon, apply it, and then we could see that we had different levels of injury and the beating up after different pressure rates. Importantly, there's a fair time delay, so you can see that directly after the injury, it actually seems reasonably fine. You can see a little bit of changes after 5 hours, and after 24 hours, it's really dead. The good thing about that, there's such a large period of time. This is the point where you want to start putting some drugs to your axon if you want to prevent it from degenerating later on down the track. So there is a window, a therapeutic window. There is a time window where actually, if we have the right pharmaceuticals and we manage to get it to our axons, to repair them again. So if we, rather than just with normal light microscopy, we look at them, you can see that this is how the healthy axons look like. And you can see the lines are all straight and nicely connected. And here you can see 24 hours after injury, you can see that the beads coming up, sounding as a sign of disease. And if you stain the cells with it, again, you can see here cell body, so it's getting all the way into the soma compartment. So we apply the injury all the way at the axon, 500 micron away. But you can also see that after 24 hours, actually the part where the cell bodies are getting sick as well. So the injury doesn't only affect locally where the injury has been applied, it gets the information, we're in trouble, mayday, we've been damaged, gets actually communicated all the way through the neuron. So we were very pleased here that we could actually use the quake valve. That's easy to make because the instructions we can just download from the researcher's website, and we can integrate that with a neuron culture device that we can either make ourselves or just buy in a shop to simulate stretch injury with unprecedented control and precision. Our results suggest that it's suitable to studying TBI, and a little bit later, we've also applied some drugs to actually repair the neurons. But we then had another student starting, and we wanted to ask another research question. And that was, can neurons actually, how smart is a neuron? And we want to get them to play in a maze. I'm not sure, are mazes popular in India? They're not popular in the Netherlands. The Australians absolutely love them. And I think it's a British thing, so that's why I thought that they may be around in India as well. So as a kid, you go to a maze and you try to go a path, and at a certain point it's closed, so you need to find your way back. So there are a couple of possible itineraries to walk. There's also puzzles that you can buy with a maze where there's a whole range of lines and you have to find your way out. So our research question became like, can neurons actually get through a maze? Can they find their way through a complex structure where there's a whole range of different obstacles? The reason we asked that question, well, officially we first started asking over coffee how smart neurons were. Then we said, let's go and try and put them in a maze and see what they do. And then we talked with a neuroscientist and said, well, if this is successful, we actually can get them to go lost or not. What could we do with it? So neuroscientists always have problems that they don't know how to find the answer. And the research question that our neuroscientists have been pondering about for a very long time is how do different populations of neurons communicate? Can they only communicate when they're in direct contact? Or can they also communicate when they're not in direct contact, but when there are some supporting cells present? So there's a cell group called the glia, and there's a couple of different ones in there. Astrocytes are a part of them, and they're always sitting around neurons, looking after them, making sure they're happy, stabilizing them. So they're kind of a support cell. So we've got the neurons do the hard communication work, but there's always the astrocytes and a whole range of other glia present to just make sure the brain is all happy and working well. So the question that we want to ask, well, if we could make neurons get lost and then confine them to an area, that device would allow us to ask the question, if we've got two separate neuron populations that are only connected through glia, if we do something really bad to these guys, do these guys notice? Because if our answer would be yes, then it means that these glia, these astrocytes, play a critical role in communication. If the answer is no, then it is you need direct contact between different neurons for them to understand what's happening. So we thought that was a really cool project, and our first curiosity was, can a neuron find itself a way out through a maze? In order to start studying that, we first needed to do a little bit of fluidics, which is when we get our food dyes out. I've seen in the little shops yesterday, India is fantastic for food coloring. I've seen the most brightly colored cakes. So if you guys will do any microfluidics, you're not going to be short of food coloring. I haven't seen cakes that were that brightly colored in a very long time. But here we got some green, red, and blue food dyes, and we made a microfluidic device with different compartments. So we had our first compartment designed to have our first neuron population, a second compartment for the second neuron population, and then here a place for the glia to be happy, to be the functional. The other thing that we wanted to do is make sure that if we would be pestering these neurons, the best way that we do it is with a chemical. To do a proper experiment, the chemical that you add to this population shouldn't end up there, because then you can't say anymore, is it communication or is it direct damage? So we have been playing around with the flow resistances to try to ensure that there wouldn't be any fluid transport from the neuron populations through the glia populations. We've talked a little bit about siphoning yesterday. To do hydrodynamic injection in CE, we've talked about the importance of keeping your reservoirs levels in microchannels. Here we did the opposite. Here we decided not to have them level. Because if we have these reservoirs here filled for the glia, and these ones with the media to keep the neurons happy, but we have the two outlets sitting above the maze empty, then it means that we get a very slow flow of our glia media through the maze to that outlet, also a small flow of our media, of our respective neuron populations to go there. But because we've got two flows going up, we don't have any flow from this neuron compartment there. So that means that these two neuron populations are fluidically isolated. There's no possibility of leakage of any of the chemicals from here to there while we're doing the experiments. So then it was time to get busy, which is, we're really grateful. So we had Ching, and I know that I've got plenty of time, so I can tell you a little bit more about the fun in the lab around. Ching did her research project with 4 supervisors. So there was Anna and Tracy were neuroscientists, Michael and I were chemists. 3 of her first supervisors in 1 year got a baby. So Ching was incredibly resilient and managed to navigate her way through all these supervisors disappearing for the various periods of time because they were having babies. And she still actually did an excellent job on a research project. She then liked working with us so much that she did her PhD. After her PhD, she stayed on for a postdoc, and she's now had 3 babies herself. So that's kind of to stop our research flow. So some of this research has taken really long. We started this in 2014, and the paper that I'm now talking about has just been submitted. Reason for that is that Ching kind of got back to us and kept having 1 baby after another and going a year on leave. But we can't blame her because we all did it to her when she was still a very young student. So that's sometimes research just takes very long because you need a pair of very skilled hands. And Ching is really unique because she's really good at culturing the neurons. Because the neurons that we work with, you need primary neurons. Who is the medical background, medical person here? Primary neurons, who knows what it is? No neuroscientist. The primary neurons are the neurons that aren't properly developed yet. Where you get them from is an unborn embryo from a rat. So before Ching can start doing her work, she needs to make sure she gets her hands on a pregnant rat in the animal house. She then needs to get the unborn babies out of the pregnant rat, then get access to the brains of the unborn babies. You don't think that are that small. Then put them in a reagent, in an enzyme solution to dissociate the neurons from the tissue that they're in. And you need to be very gentle because neurons don't like being stretched because if they get injured very easily. And once they're dissociated, that's the point where you can actually start loading them into your devices and do experiments with. So whereas normally as an analytical chemist, life is good. You make your solutions and you start in the morning. Ching is already about 2 weeks into an experiment before she actually gets to the microfluidic device because she needs to plan the rats for surgery. She needs to harvest, dissociate, stabilize, culture up the neurons, put a whole range of reagents to them. This is really hard work, and that's why it's so important to work with people in medical research because they've got the infrastructure and the know-how how to actually get to primary neurons. Because if you're with a surgery, it's actually quite easy because you take a pair of scissors. But I wouldn't have known where to start to get access primary neurons. Where do you get them from? How do you get them out of a rat? How do you find baby rats in the belly of an adult rat? So there's a whole range of extra skills if you want to do cell culture that you need to get as a microfluidic researcher. So it's very good to partner up with someone in medical research that already has got all those skills because it takes ages to develop them yourself. That is a side note. Neurons aren't very smart. To our great delight, we put our neurons in our banks and we went through a couple of test structures to just see how far they could go. What you can see here, we've got our neuron compartment. So here, this is where we see the neurons. We've got different banks here, different rows of obstacles. You can see the neurons are slowly growing in. See an obstacle, think, ooh, that's scary. I make a U-turn and I get back. Some of them accidentally get through the first gap, second gap, third gap, but you can see the number of neurons that actually find their way through is very quickly dwindling. And if you look, the first one, we still get about all the neurons get through the first and second, but it very rapidly decreases.
In which medium they go?
Culture medium. So it's got a 4-letter abbreviation. So it's the standard medium that they use to culture the neurons in Petri dishes as well. So you can see the neurons squiggling around. And then by the time we're at banks 9 and 10, there's no neurons coming through anymore. So if we make a structure that's got 11 or 12 different banks, we can guarantee that no part of the neuron will find itself into the glia compartment. Next thing we did is trying to culture the glia. And glia are very easy to culture. They don't have long arms and they form carpets quite readily. So you can see that we seeded the glia into the astrocytes into the astrocyte chamber, and they quite happily moved through, they replicated themselves, and they filled the whole space up. So whilst the density of glia was a little bit lower at the end than it was at the beginning, it was very clear that it was a matter of time and we would have a perfect, nice red glia carpet all the way through. We then combined the two of them together to start doing our experiments. And what we did as a pestering thing with the neurons is add some cyanuric acid, which is a toxin. So here we've got the device where we've got the axon population seeded where we apply the injury to. This is the neuron population that we want to see if it notices what's going on. And this is where the astrocytes are that hopefully form our communication medium. And to our great surprise, so we can see here the red ones are the left culture compartment where we apply the injury. The gray bars are the other sides. Here we can see that if we apply an injury, we can see some unhappiness, some bleeding happening in the left compartment. But to our great delight, we could actually see the same happening on the right hand side. So you can look here, this is the control, the vehicle is just adding some water or something that looks like the solution of the cyanuric acid, but does no harm to the neurons. So this is our control. We can see that both the left co-culture compartment, where we did apply the injury, and the right look the same. Whereas if we apply the cyanuric acid, we can see that the left side, where we apply the toxin, shows bleeding. But the right side, where we haven't applied any toxins, actually starts really showing that it's really stressed as well. So that shows that it's our glia here that cause the communication. Because if we do a repeat where we don't have our astrocyte culture in, we do see the injury in the left-hand side, but we see very, very minimal injury on the right-hand side. So we can conclude now that the astrocytes here play a role in neuron-to-neuron communication, and there's no other people yet that have been able to do an experiment to prove that conclusively. The next question that our neuroscientists wanted to ask was, so how do the glia communicate? And the hypothesis was that calcium was going to be the communicator, because calcium is a great chemical that a lot of cells use to communicate. So what they wanted to do is to add a calcium chelator. That means that whenever there's calcium being released by the glia, it's actually mopped off by the chelator and it can't be going to the next cell. And indeed, whenever we added the chelator, which was abbreviated as BA, the difference in injury response was erased. Unfortunately, there was some toxicity effects of the chelator itself as well, but we could show that we have not only now demonstrated that the glia, the astrocytes, play a critical role in communication between neuron cell populations, We've also demonstrated that it is calcium that is used as the neurotransmitter to transmit signals between glia. So we've just submitted a paper. We have really enjoyed that as a project where they had a serious need. They had a problem where they wanted to answer a research question that they couldn't because they didn't have the tools to do so. And we could play around making cool structures in the lab and actually help our neuroscience friends to come to great results. So working together with neuroscientists or any other medical researcher allows someone that's in the area of microfluidics to enable experiments that have not been able to be done before. And yeah, it's a really cool thing. It's not an analytical use of microfluidics, but it's still very nice. And if you've got a little bit of a biomedical interest, hopefully you can see the potential of what very simple microfluidic devices, because these things weren't hard to make, what a difference it can make to medical research. So that was the little bit of information about what you can do on cell culture that I wanted to tell you about, considering we have a little bit of time to fill before Professor Bagat arrives here.
He will start at 2 o'clock. His timing is 2 o'clock. As per the schedule. You want to talk to me?
So, yeah. Well, let's break now for coffee and then for tea.
At 11.
Yeah.
Okay. We can have a little bit break and we'll reassemble after in 11 past. Either my own section with something or EL of EL. Also EL is like punch. Monday food.
Well, I think it leads with him that he would do all the simulation contents. So I've also he thinks that we'll do that problem.
Never come. You can see the process.
I've seen the workshop, but we then communicate because he was. And Do you maybe use read card for me? The file is on my hard drive and not on the internet, so do you have a USB card for me? So I can put it in?
Well, You can copy it and
Yes, sir. You can
internet ला देना dongle ला
So electron membrane extraction is another area where you use the affinity of a membrane for a certain analyte that's driven towards your membrane given the electric fields. Doing that on a chip, and we'll hear a lot more about integration of porous materials and because filtering on a chip has been a passion of mine for a very long time. Since making this presentation, we found a way to 3D print nanoporous materials. But at this stage, we said that the integration of the porous material on the chip is really, really challenging and hard. Is there a way to create porous structures in a chip that's already made, not having those pores in them? And we then started thinking, can we use lightning at a very small scale to break down a material by dielectric breakdown to create nanometer scale pores? And that's some of the work that a PhD student of mine, Alia Shalan, has done. So what we did was applying a high potential difference between a V-shaped microchannel and a T-shaped one in PDMS. And it's my laser pointer. There we go. So that's already all experimental data. So what we did by applying a potential difference, so here you can see, and then don't look at the colors yet. Here you can see that we've got a microchannel and a V-shaped structure. What we did is apply a very high potential difference above the dielectrical breakdown of the material for this 100 micrometer wall. And with that, we could control the breakdown of the material sitting between this V-shaped channel and of the V-shaped chamber and this microchannel. We could control the speed of the breakdown by controlling the current during the breakdown process. So we initially had, already during my PhD, I'd seen that if you apply too high a potential difference across two microchannels, at a certain point you get sparking and you get the dielectric breakdown of the materials in between, because every material has got a dielectric breakdown constant. And if you bring your channels very closely together, you actually don't need extremely high voltages to electrically break down the material. So we played around a little bit with that, or Alia did a great amount of work there. And we figured out that to control that breakdown process, you need a very high potential difference to make that initial connection. But all the damage being done after that is a function of the current density. Because the current generates the heat, and it's typically the heat that melts the materials and creates the damage during dielectric breakdown. So what we did here is use a high voltage power supply, but put a current limitation in place, so that as soon as the current established, we could actually drop down the voltage so that we had a very small current, and with a smaller current create smaller pores. So here you can see how effective that's been. We can see in here the current limitation that we put on our power supply. So we allowed it to apply a potential difference higher than the dielectric breakdown voltage of PDMS over 100 micron. And if we applied a restricting current of only 1 microamp, we could demonstrate that if we then clean the device, open it up, and apply the potential difference again, we filled one of the chambers with iron, and the other one with thiocyanate solution. We could actually, when we apply the voltage, see the representative orange color of iron thiocyanate. Transport from iron and thiocyanate through the pores that we create through dielectric breakdown. However, if we put in some rhodamine, which is a small molecule dye, rhodamine has an extra mass, from memory somewhere 350 So it's small organic molecule, but it's not as small as iron and thiocyanate would be. So we did the same thing, same device here. It's black because it's fluorescence microscopy that we use to visualize it, because there's very few things that are as nicely and brightly colored as iron thiocyanate. So potential difference got applied here, and you can see that the rhodamine 6G didn't get through the pores. So that means that the pores, when we use 1 microamp breakdown current, the pores were large enough to allow for the transport of iron cyanates. However, rhodamine 6G was too big to fit through. So we had very, very, very small pores. When we applied slightly higher currents, we could actually get rhodamine 6G to pass through, but carboxynephtalfluorescein didn't want to go through. This is a little bit of a trick in the charge of the materials, because we are talking about very small channels here. We've heard Professor Philip talking about the electroosmotic flow, and we had the negative surface charge, and then the fixed layer of cations that sat on top, which also had an associated fixed layer of anions. And then we had the mobile cations that were the ones that started moving to drive our electroosmotic flow. He had all these pretty diagrams in there. That kind of looked like that, where this was the distance from the walls, and there were molecules, and there were molecules increasingly far away from the wall, and then there was the Z adaptation. All these ions, so if we've got our negatively charged surface, and we then have got our immobile cations, our immobile anions, and then we've got our mobile cations that drive our EOF, they all have a size. And that size doesn't really matter if the other wall here that's got negative charge is more than a micron. Doesn't matter, because physically this wall can't see that wall. If you go to really, really small pores, and in this work we actually are talking about really, really small pores, because we knew at 1 microamp current being applied, the pores were small enough to allow iron and thiocyanate to get through, not rhodamine 6G. So these are pores that allow molecules to pass, but also have the ability to block molecules. If the pores are the size of a molecule, that means that our other wall is actually sitting here, which then ends up that we get a problem with electron-repulsion. So we've got the cation layer here, that is the first mobile layer. So if we've got a negatively charged surface, in this instance, rhodamine 6G, which is a cation, can take that role and still find its way through. If we are talking about the fluorescing equivalent, which is negatively charged, we get a repulsion effect by this negative surface charge, and it's sent away. So we end up with a perm-selective core here that, because of its negative surface charge, PDMS by itself is negatively charged, it repels carboxynephtalfluorescein, but it allows the cationic dye rhodamine 6G to go through. If then we increase the pore size, it's large enough that we've got a little bit better balance of cations and anions that are swimming around in our pore, and the carboxynephtalfluorescein is no longer blocked. However, BSA, the albumin protein, is a lot bigger, because carboxynephtalfluorescein is a little bit larger than fluorescein, so it's probably 550 Dalton. BSA is a couple of kilodalton. So BSA is a really big molecule. So at 3 microamps, we didn't have any charge-based repulsion anymore, but BSA couldn't get through. And if we then increased our currents to 5 microamps, the BSA actually could get through our pore, but if we injected the diluted blood sample, the cells in the blood stayed behind. Which means that we can filter. That is exactly what we wanted to do, because that means that we can get proteins and any other smaller molecules, because the pore size is only restricted for a certain upper value. We could extract molecules from whole blood through the pores that we created by dielectric breakdown. So we found out that the transport is based on the charge and the hydrodynamic radius. Double layer effect is responsible for the cation and ion selectivity. And the co-ion, so the ion that's got the same charge as the surface, is the one that notices the repulsion. What we use this for is the detection of quinine, which is present in tonic. It's also used as an antimalarial from whole blood. So you can see that here we put a whole blood sample. As you can see here, we put a blood sample on one side of our ion mobility trap, of our dielectric breakdown features. And here we had a channel where we could do electrophoresis in. And as you can see here, we get increased peak height when we increase the concentration. Our migration times here are not completely repeatable yet, but that's because we had some problems in timing the injection. So that was more so the starting of the recording. But we had a very nice correlation between the quinine concentration and the peak height. So from here, we were very excited, delighted, got a great publication out, but we thought that we could do more. And that's when we started thinking, we came up with the concept of the ion mobility trap, because we had the possibility in hands to selectively allow for the transport of some molecules, but not of others. So what we wanted to develop is a device where we could put a very dirty sample in on one side, then allow molecules below a certain cutoff to get through the holes here. So the big blue and red blobs here are too big to fit through these holes. They're blocked, like we've seen in the blockage pictures that I showed on the first slide. With our target analytes, that may actually mean that small inorganics like salt, anyone that has been working with biological matrices knows that blood and urine and everything, they're full of salt. In electrophoretic assay, you don't like high conductivity. So actually, in a lot of instances, we want to desalt, we want to get rid of some of the sodium chloride that's surrounding our sample. Which is why here we've made much, much smaller pores that only allowed for the passage of these small inorganic molecules. Which then means that our target analytes, that were the small organics, would be remain in our microchannel. From a dirty sample, we could leave all the large molecules behind, and we could even get away from the small molecules, the small inorganics, and desalt our sample. We use this one for the concentration of fluorescein and eosin, and you can see that here we use the pinched injection, and we've been through the pinched injection yesterday. So if we use this sample with pinched injection in a normal microchip, this blob here is the peak height. Can't even see the eosin, but here you can see the fluorescein peak. Hardly there, and just within the baseline noise. If we use the size mobility trap, where the fluorescein here concentrates, we can get up to 100-fold concentration enhancement. Reason that we get this concentration enhancement is that we allow this electric field to be applied over an extended period of time. So we apply our potential difference. Fluorescein is negatively charged, so it likes going to the positive electrode. So we apply a positive potential to this reservoir, a negative potential here, and we just keep it going for a minute or so, which means that our anions, our chloride, as well as our fluorescein, are continuously extracted from the sample reservoir migrate in the electric field to the entrance of this channel. The chloride, negatively charged and very small, zips through these pores and goes into the buffer reservoir. The fluorescein, however, is too big, so it wants to go through the door. It's collecting, concentrating here in front of the door, because in the it wants to move in the electric field. It wants to move to the positive electrode, but it can't because its access is physically blocked by the membrane. And that's the concentration enhancement effect, because continuously as long as our potential difference is applied, we get the transport of fluorescein from our sample reservoir into the microchannel, and we get concentration enhancement, factor 100 concentration enhancement here, because the fluorescein stays in the electric field. It wants to go towards that positive electrode, but it can't because it's blocked by a membrane. And so there you can see that that tiny little blob actually indeed was a peak. So this system allowed us also to extract drugs from whole blood. So we worked with the antibiotic ampicillin, and here you can see that so we did some labeling of the ampicillin to get fluorescent signal. There were some other compounds, primary amines in the blood as well, that labeled. So that means that our blank signal, the experiment that we did with blood without our compound out there, still's got some peaks. So our blank is not a perfectly flat line. We've got some other molecules in the sample that are small enough to fit through the first pores, big enough to be blocked by the second ones, and therefore show up in our signal. Luckily, however, we did after concentration, we did an electrophoretic separation. We did some zone electrophoresis, and that allowed us to separate out the ampicillin from the interferences still coming along from the blood. And again, happily, we had a linear correlation between our concentration of ampicillin in the blood and our peak height. So we could quantify our ampicillin concentration in the patient's blood really quickly. So this is when we talked about commercialization. This is the device that we thought was going to be great in the treatment of sepsis. Because one of the problems in sepsis is that the pharmacokinetics in the body go completely haywire, because all the vesicles get leaky. So the standard calculations for the administration of drug dose don't apply anymore. And you kind of need to put some drug into the patient, measure the antibiotic, and see if your the concentration of your drug is actually within the therapeutic window. Because quite often, because the distribution volume in the patient increases a lot, the effective drug concentration drops during sepsis and is no longer effective, and then the patient dies. However, when we talk to an angel investor to get the money, we were told that saving lives is actually not a priority, because once people have sepsis and are that far advanced, if keeping them alive creates a significant medical bill, and insurers don't like to pay that. So this is the little bit of work that I refer to that we thought was going to be a great project, great product to have in any emergency departments, and we were told no. But we were very excited about this work, but did realize that the applications were perhaps a bit limited, because there's only so much analyte you can actually, in a unit of time, push through a pore. So we were limited by our concentration sensitivity, because at a certain point, electrolysis started playing a role. We did try to change our V shape, our little tip, to a U shape to get more flux, but that wasn't really successful. So we started thinking about other ways to put get more flux through the membranes and still build our ion mobility trap. So what we did is integrate some trick-edged membranes to get the same effect, still construct an ion mobility trap, but because we had, instead of only 1 or 2 pores formed by dielectric breakdown, we'd have thousands of pores already coming through the membrane. So the way that we made these devices experimentally is that we started off creating a microchannel. So we did this by casting it in PDMS, and we had a nice, very simple mask, simple channel, about 5 centimeters long from memory. We then put 2 bits of membrane in place, and Fung, the student working on this, was really, really patient. So he cut by, I think it was 2 by 2 millimeters, bits of the track edge membranes with really sharp scissors. Really struggled doing that because they shouldn't be bending on the edges because you wanted to have them flat sitting on the chip. But Fung was very patient and managed to get the 6 micron thick track edge membranes on there. And we bought 2 different ones off the shelf. We bought a track edge membrane with an average pore size of 10 nanometers and one with 100 nanometers. So poor Fung had to cut these tiny little squares from the track edge membranes without curving and nicking the edges for 2 different membranes, and we stuck them on top. Next thing was to come up with a second PDMS slab that again contained microchannel, but this time it contained 2 microchannels. 1 microchannel that was sitting on top of the 100 nanometer pore size membrane, and 1 that was sitting on top of the 10 nanometer sized membrane. So fairly tedious microfabrication process, but it required a lot of patience and resilience from Fung as a student. It didn't necessarily require very expensive stuff, because one of the things when both now and the University of Tasmania, we didn't have a lot of advanced microfabrication facilities. This will make sense to next week when we've talked about microfabrication, but we could do some spin coating and making templates ourselves. And we could do some work in a dry film photoresist. That's the devices that I passed around earlier. But compared to a lot of universities in the world, our microfabrication facilities were really, really limited because we didn't have a microfabrication, we didn't have a clean room on campus. We didn't have access to a clean room, and we didn't have the budget to pay someone to make make devices inside a clean room, which is an alternative. So to use these devices, we wanted to do electrophoresis again. So we started working with our favorite anionic dye fluorescein. So we applied minus 500 volts, which allowed our fluorescein to move through the 100 nanometer pores and then up to the 10 nanometer ones. And then they were concentrating there after having injected our sample through the 100 nanometer pores and being concentrating at 10 nanometers. We could do a normal injection where we applied 1200 volts to the end of our separation channel, minus 300 volts there to balance the books, small pullback voltages here as we talked about yesterday, and we could do an electrophoretic separation in our microchannel. Picture's always good. So first the concentration of the fluorescent dyes while we were doing this. So this is the analyte that is concentrating at the edge of the track edge membrane. You can see the dashed lines here. They reflect the microchannel, this horizontal microchannel. That's the line that we're looking at here. You can see that this concentration happens a little bit offset, and that's because the track edge membrane's got a lot of holes in there, and there's actually already some fluidic connectivity from the very edge of the membrane to the microchannel. And the very first point of electrical connectivity is where the concentration takes place. So that's why we're slightly offset from our reference channel. So here we can see that we worked with a natural fluorescent protein in TBE, which is a standard buffer. And here you can see the concentration of our phycoerythrin, 50 seconds, 90 seconds. You can see the concentration being enhanced. And once we thought that it was enriched enough, we could do zone electrophoresis. And here you can see the migration of the R-phycoerythrin in the microchannel. And you can see that we could get fairly good concentration enhancement factors up to 1,000 for a highly dilute solution of 0.2 ppm. Because of all kinds of repulsion effects, the higher the concentration of the analytes that we had, the more modest our concentration factor was that we could achieve. So this was really good for low concentration and low concentration samples. Not that good when the concentration went a lot higher. But we did also demonstrate it for a separation, and we used here our phycoerythrin, the red protein that I showed before, and labeled BSA, which is blue here. So you can see the concentration enhancements taking place here. 30 seconds, you can kind of see the blue and the reds both concentrating. Then after 70 seconds, you can see a really big intense band being formed. And if we then start the separation, you can slowly see the faster migrating BSA moving apart from the R-phycoerythrin. So these are photos looking on top of an electrophoretic separation after having done the pinched injection. So we're after 20 seconds, we're sitting around here. So you can see that the BSA just is almost baseline resolved from the R-phycoerythrin. Here on this image, you can see that the R-phycoerythrin really lags behind. And we've got an area where we just have got background electrolytes before we enter with BSA. So that's what's happening at 30 seconds. So we were very pleased that we could get up to 900-fold enhancement in BSA concentration when we used a 10 ppm solution of BSA and 2 ppm of R-phycoerythrin. So you can see that we get really nice concentration enhancement relative to an absolutely tiny signal when we just used the normal pinched injection. They are the wrong way around on that slide. We tried applying this for an application, and we thought about microalbuminuria, which is the problem that your kidneys don't work really well anymore, and you have protein that starts showing up in your urine. So Fung applied this to a urine sample where he spiked in 5 micrograms per milliliter of human serum albumin. So normally BSA, we use the bovine version. In this case, he was working with his own urine, and we put some human serum albumin in. Doesn't make too much of a difference. And there is a tiny amount of albumin present by itself. So you can see that the intensity by spiking the human serum albumin in, you can see that the peak height increases by using the size mobility trap. And here you can see that we had a linear correlation between the amount of serum albumin spiked into the urine and the peak height. So we were able to quantify the amount of serum albumin in the urine sample, which is a very important early diagnostics for kidney failure. Okay, changing gear and going a little bit closer to what we're going to do this afternoon. And we go fishing. Anyone done fish? Yeah, so that's what we try to do in an electric field. So we've talked already this morning about isotechophoresis and the way that we've got a discontinuous electrolyte system where we've got a leader electrolyte and a terminating electrolyte. The leading electrolyte has got an ion that's got a higher electrophoretic mobility than the terminating electrolyte. It also means that that solution has got a higher specific conductivity, and that means that its electrical resistance is lower. If we apply a potential difference, we get, because nature wants to have a constant current going through the system, we get auto-adjustment, auto-regulation of the electrical field strength, which means that in the leading electrolyte, where we've got high conductivity, we've got low electrical resistance, and therefore we get a low field strength. In the terminating electrolyte, we've got lower conductivity, and therefore we get higher field strength, which is depicted here exactly the way that we talked about it on the board. We get a drop in ITP in our electric field strength, moving from our terminating electrolyte to the leading electrolyte. Isotechophoresis, we've talked about it's normally done in the plateau mode. We didn't say it was normally done in the plateau mode. I left some detail out, and I told you that the length of these zones is dependent on the concentration. That's again also depicted here, where you can see that the concentration of the blue ones is lower than that of the green ones. The red ones are present in lower concentration as well. So we get our analytes, they rank themselves in terms of mobility. The reason that's an auto-adjusting, self-focusing system is if we've got the green analyte here, that gets really cheeky and starts running a little bit faster than it should. It ends up here in the zone filled with the red electrolyte. However, the field strength is a bit lower. It accidentally ends up here, oopsie, field strength is lower. It's got its own mobility, can't go faster, which means that at its mobility, right. So brakes go on, it goes back to where it belongs. Same thing if something would happen and an analyte from the green cluster here somehow gets stuck somewhere, gets retarded, ends up in the blue zone, then all of a sudden it finds itself in a much higher electric field. Based on its mobility, whenever it's exposed to a higher electric field strength, it zips along and it goes back to where it belongs. So it's a self-focusing system by isotechophoresis. So here we've got direction that the ITP is moving, electric field is being applied, and you can see terminating electrolyte, leading electrolyte, and all the sample ions separate and order according to their electrophoretic mobility. This afternoon, so this work comes from the group where Professor that will be here this afternoon will be a bit, I don't know how long you worked there, but certainly a couple of years and some great work. If you're interested in this, Professor Santiago has published some absolutely fantastic work. I don't mind the music, I quite like it. So the Santiago group did absolutely some fantastic work. All these papers take at least 10 times of reading before you understand them. So don't give up if you don't understand them after the 5th time. It's really good stuff, but you really need to think and draw things next to what they do. The concept that they cornered and that we used for the fish staining, that's why I'm dwelling on it for so long, is the peak mode ITP. Because we've just been talking about adjusting concentration, having different field strength, having self-adjusting, self-focusing mechanisms. All works well and good when you've got plenty of analytes to go by. Sometimes your analyte is present in a very small concentration, and it actually can't reach a zone that has enough conductivity to sustain that field strength, which then gets a crisis in ITP, because you want to have that gradual drop. Nature wants the same currents. We can't have a pile-up of electrons. So we've got a current through a system, and all of a sudden we've got nothing to carry that current. We can't all of a sudden have a build-up, a traffic jam with all electrons beeping their horn like in Indian traffic. We will have chaos. Current just needs to flow. It's like water. We don't want blockages. So we want to have that current flowing through a system. We want our system to all move with the same speed. But all of a sudden, if we don't have enough of this analyte, this red analyte here, we can't get the conductivity to carry the field strength. And what then happens is that the two different surrounding zones, which could be the terminating and the leading electrolyte, it could also be two adjacent analytes. They really jam up together and they help that area that is in serious trouble to still get to the conductivity required to carry that field strength, to carry the currents, and to allow the isotechophoresis to move the way it is. And by doing that, it really squashes the target analyte that's not present in enough concentration in a really narrow zone, because it wants to get up to that conductivity. So you want to have all your ions pretty much lined up on a single line in your ITP system to carry that field strength, even if it's only on over a nanometer length. And that phenomena, while it's a crisis in electrophoresis, it's absolutely great and fantastic as an analytical chemist, because it means that our trace analytes, we can focus in a really narrow zone. All our detections are concentration sensitive, so we can get much lower detection limits because we can sweep up our analytes in a very narrow zone, and that's called peak mode isotechophoresis. Then we've got normal isotechophoresis, where we have our leading electrolyte. We've got our terminating electrolyte. Our leading electrolyte has got a higher conductivity and therefore a lower electric field strength. Terminating electrolyte, lower conductivity, higher electric field strength. We get our analytes to adjust their concentration in zones so that we get a slow drop in our electric field strength, so we get a continuously moving system. Isotachophoresis, same velocity. So we adjust the field strength here. Peak mode ITP, our concentration isn't high enough to get a plateau, and what actually the analyte does is do everything that's within its power to come to the level that it needs to do to sustain the locally dictated field strength. But you can do with that as well. So as an analytical chemist, that is great. Isotachophoresis, peak mode isotachophoresis is used in so-called transient systems where we do a little bit of ITP and we then dilute and do another trick with the background electrolyte. So we end up in zone electrophoresis again. But we've got all our analytes very nicely focused and concentrated together. What the Santiago group figured out as well is that hybridization, the binding of dyes to DNA, is a concentration-driven process. Brilliant thought, not really rocket science, because we all know that the equilibria are dependent on concentrations, and we get complexes to form faster if we've got higher concentration. What was really cool though is that they demonstrated that you could use peak mode ITP to significantly accelerate hybridization of DNA. So here we've got an ITP system moving at the same velocity because it's ITP. We've got a trailing electrolyte and we've got a leading electrolyte. And we put trace levels of two complementary strands of DNA in there. We apply a potential difference, and therefore we get an isotachophoresis system to be formed. Our leading electrolyte still's got high conductivity, low field strength. Terminating electrolyte, lower conductivity, therefore higher field strength. In the middle, because we've got only small amounts of DNA there, we've got our little crisis happening and we've got our peak mode ITP. We get our very concentrated focused band because our concentration of our two DNA strands is actually not high enough to carry the current that's required to sustain the electrical field strength. So we get a little bit of help of our terminator that's tailoring off here. We get a little bit of help of the ions from the leading electrolyte, and it's really squashed in a really, really nice tiny band. Which means that we get our two complementary strands, strands A and B, focused in a very narrow zone here. That interface, that characteristic interface, how close the terminating and the leading, that's a fixed number. You can calculate that, it's in the papers. And impressively, there was a 10,000-fold enhancement in the hybridization speed, doing it by ITP than when you would be doing it in free solution. 10,000 times faster. That's worth spending 5 days on the Santiago paper to try to get your head around ITP. So we'll get some help with that this afternoon as well. But that's the kind of power that you have when you can use an electric field to really, really concentrate things so that they are in close proximity. By the end of the day, molecules, reagents need to be in close proximity in order to react. One of the things peak mode ITP can do is bring things in very close proximity, provided that their electrophoretic mobility sits between the leading electrolyte and the terminating electrolyte. So our idea was, can we actually do that as well to put some probes into cells? So we had a capillary that was filled with a leading electrolyte, a trailing electrolyte. We had some cells in here, a probe, and something called a spacer ion as well, in order to separate different fractions out a little bit better. But yeah, I don't think we need to talk about the spacer yet. So what happens whenever we start working with the ITP? We have our probe that had a slower mobility than the leading electrolyte. It comes out. Our cells that were faster than our terminating electrolyte, or even a little bit faster than our spacer, came out as well. They were at a lower concentration than the leading electrolyte, trailing electrolyte, and our little spacer, which is literally something that gives you a little bit of more room to move and play with. So we were able to put the cells and the probe in very close concentration. Great thing is that very high concentration of probe forces the probe into the cells, whereas staining normally may need a lot of energy or electroporation. If you actually locally have a very high concentration of probe around your cells, the fish probe just enters E. coli by itself. So that's what is happening here. We have our cells, we have our probe penetrating, and all of a sudden we get fluorescent cells. Next thing is, because we're measuring fluorescence, is that we needed to get rid of the free probe, because the probe is fluorescent both inside and outside the cell. The way we did that is, in addition to having our electrolyte system with our leading electrolyte and our trailing electrolyte, and I sneakily didn't tell you about it, we also have a sieving matrix here, a little bit of a polymer that works as a gel. It's sitting here doing nothing while we do the hybridization, but isotechophoresis, it's all moving. Isotechophoresis, same speed, it's a dynamically moving system. So this ITP zone where we've got our cells, our label that has entered the cells, but maybe some excess label as well because we want to have high efficiency. When that hits our gel, then the stained cells and the non-stained cells are being delayed because it really struggles to get through that force of the sieving matrix. At the same time,
The all electrolytes are stationary.
no, the electrolytes move. So the ions move through the gel. So the gel
Plugs like one, first, second, third.
Yeah, so the gel is stationary. The ions migrate through the gel. So the
leading left the first one, second one, and third one, the trailing. All these are stationary at their own positions, or they're moving?
they're moving.
They're moving.
So the
Leading electrolyte in gel.
Yeah, so the gel is made up initially in leading electrolyte. Then we also have a zone in front of that that's leading electrolyte without gel. Then we've got our sample. Then we've got our terminating electrolytes. All the anions, so we're talking anions here. What are ITP systems? Call it terminating minus, leading minus. They are all moving with the velocity of the ITP system. So the ions are all moving. This window moves to the gel. The gel itself is neutral, so it doesn't move. We have no EOF system. So the gel stays here, but the ions, we've got the leading electrolyte here. In the field, this ion moves through to our waste reservoir. So just like gel electrophoresis when you're looking at the DNA gel, where your slab gel stays stationary, but your DNA moves through, and your electrolyte ion moves through as well to carry the current. It doesn't care that it's in the microchannel. The gel sits there, it's stationary. The rest of the analytes have charge, and they move through. And the gel is permeable for all small molecules, just not for the big blobby cells that we've got. Good question. And then it's now clear why we had that spacer in there, because we wanted to have, so we have our cells being delayed, but we wanted to very much separate our free probe from our stained cells for easier quantification, so that we'd have a very nice and focused free probe band that we could detect first. Then our spacer, that's neutral, so our detector, or that's colorless, so our fluorescence detector wouldn't see it. And then we've got our stained cells coming through. And that worked fantastically. So this was not done microfluidically, this was done in a CE capillary. Here you got the leading electrolyte is a tris HCl. So chloride is the leading electrolyte. We stopped the physical flow, so our migration is, all our transport is based on migration. Tris Hepis is the anion spacer was messed with a bit of tris, and the sieving matrix was 1.8% hydroxyethyl cellulose. So that's a reasonably viscous solution, but that's still, viscosity is low enough that you can still, with hydrodynamic injection, load it into a capillary electrophoresis instrument. What you can see here is that we get a big peak of our free probe. Our free probe was coming first. So that migrates first. So if we look here, we've got our free probe, a window, and then our cells. So we've got our free probe, a window, and then we can detect with a fluorescence detector our stained cells. And we can see here that our peak height nicely increases with our cell concentration. So we can do staining of E. coli cells in a capillary electrophoresis capillary by using peak mode isotechophoresis. And we could do the same thing if we change the fish probe. We could do exactly the same thing for Pseudomonas aeruginosa. So with fish staining, it's a fluorophore that is DNA specific. So if you change the DNA sequence of the binding probe, you can change the target. And because all the bacteria have a relatively similar mobility, you can translate this ITP system and apply it to another bacterium. And when we then look at the specificity, whenever we use the probe for Pseudomonas, you can see we get a peak. And even if we have E. coli cells present, the probe doesn't bind to the E. coli cells, which means that in the ITP zone, the probe gets in, can't go anywhere, so it migrates out again as soon as we hit the gel, and we don't end up with a fluorescent signal. So it is a specific probe, and we have, despite doing ITP and at a high concentration, we have preserved the specificity of this analyte for the staining reaction, just as you'd expect to be doing with fish staining. And our final detection limit was ended up being only 2 of the norm, so we could see up to 3 to the 4th cells per milliliter. The legislatory limit for E. coli in drinking water, we'll look in drinking water here, is 1.5 times 10 to the 4th cells per milliliter. So we're only a factor 2 of the regulatory limits, with that difference that our ITP-based assay only took 10 minutes, whereas the normal fish staining protocol takes 2.5 hours to complete. So it's a really fast way of detecting E. coli using capillary electrophoresis if you apply a peak mode ITP approach. We did try to visualize this on the chip as well. And so here you can see the microchannel where we had some different inlets and outlets. So we can see here in the first one where the probe and the cells are finding one another. The blurbs here are the cells. And if the voltage stopped, you can see the stain ending up in the cells as the bright blobs. And the stain then still needs to be removed. So we did manage to translate the method from the fused silica capillary to the chip, but our microfabrication methods were quite limited to actually get proper enough control of putting all the electrolytes in. So, but we were very pleased here that we could actually see the stained cells. So the fish works on the chip as well, but whereas with the CE we could get really nice, quantifiable and defined bands. The zones on the chip that we could see were a little bit more diffuse, particularly when the voltage was stopped. And 3D printing for microfluidics chips is something that we'll be talking about next week. So I will interrupt this presentation that I had before, and I hope that I've given you a little bit of inspiration on capillary electrophoresis. Professor Philip has given a great example on the breadth of analyte classes that you can do separation with when you've got a capillary electrophoresis instrument. And you can use pseudo-stationary phases, micelles, to play around with the resolution. I hope I've convinced you here today that by playing around even more with the electrolytes, with the separation chemistry, you can actually do really funky things, enhancing concentrations, doing stacking, and enhance biological biochemical interactions. So Phil showed you that we can measure bioaffinity interactions by using capillary affinity electrophoresis. I've now shown you that we can also play with those equilibria by locally enhancing the concentrations and therefore do advanced biochemical assays. Like Santiago Group demonstrated, a 10,000-fold enhancement in hybridization kinetics, which means that it can make the workflow a lot faster, and it can vary standard protocols like fish staining. Using an ITP method, you can actually make it a lot faster and to conduct. So these are the kind of things that if you play with electric fields, with the membranes, with the size mobility trap, trying to concentrate them, play with your electrophoretic mobility, the direction where they want to go, and physically block that either with a sieving matrix or with a membrane, that allows you to concentrate analytes as well and get to much more sensitive results. So I believe that if you don't have any questions, we're ready to break for lunch. If you do have any questions, it would be a delight to answer them. So, any questions? I hope you're all primed and excited about doing some simulations for isotechophoresis this afternoon, because, yeah. I hope that I've now talked about leading and terminating electrolytes enough that at least you've got a good starting point for this afternoon sessions. And I'm certainly looking forward to the session of this afternoon. Thank you very much. Bye-bye.
So, I understand that we, at 2 o'clock, we will bring your laptop if possible, and it will be very interesting. Of the software.
It's free. It's freeware. So there's a statement here.
And if you want to make chips, not aloo chips.
Go. Web-based open source platform, free to use for anyone. That we'll be talking about this afternoon.
So mechanical engineering is with publishing in chemistry journal. And really, it was a great Tomorrow also will be more interesting training on field.
Again, if you start reading the paper, focus for now on how to do it and not on how to crash it right. I have no idea how to do that.
Main thing is, it is not necessary that you are working in the organic inorganic chemistry. The main thing is learn the new things. And these are the new techniques. Like now, I'm telling you, there is a competition in the market, like NMR imaging. No, NMR imaging. If you go to the market, people are asking, you cannot be chemical or with software engineers just to find out the neurons which are broken. Then one software developer will say, I can magnify this neuron 100 times. The other will say, I can make it up to 200 times. Magnify like this. And this is so much means they are making research, business, and you cannot imagine how cleverly people are working on small things. And why we are lacking these things? Because we don't have introduction to these. These are not difficult. But if you go to a mechanical department for last then I think the big chips will be very good for you. They will be also with us for the weekly lecture, and you try to PDFs for it as well same chips. PDFs is पहले peanut butter आपके पास भी बहुत PDF smartphone है and try to put these types of chips.
I'll quickly show you how easy it is to get to.
And by playing with these tips, तो दो बातें रखा हूँ ना PhD में दो बातें एक तो फिर भी जो सुपरवाइजर ने बॉस में तय किया कि भी वही ठीक है it is not like so मेरा जो मानना है ना कि we learn sometimes many things from them. It is not that because you are the people who are working on the desk. And from you, we get many points. And if you try to develop like chemistry, pharmacy, and biotechnology, I'm telling you my experience. This is a glucose, हम sugar test करते हैं. Sugar test करते हैं. Sugar test से लेके आप ये देखो कि शुगर टेस्ट की वो छोटी सी किट है उसको पढ़ाने में कितने आदमी का हाथ फिर उसमें इलेक्ट्रॉनिक्स भी है उसमें केमिस्ट्री भी है उसमें फिजिक्स भी है और उसमें इंस्ट्रूमेंटेशन मैकेनिकल भी है नहीं है मैकेनिकल है तो ये तो सारी चीजें अब कोई चीज है अब रबड़ ही है रबड़ होती है कोई कह देता अभी मुझे तो काली चाहिए मुझे गुलाब चाहिए गुलाबी चाहिए पीली चाहिए और एक आएगा तो सैंड वाली ये जो है ना बात है क्योंकि उसको सैंड वाली भी चाहिए साथ में उसका इश्यू भी This leads to anxiety. So, ये तो पॉलिश काम करते ही हो these are very, very easy. And weak eutectic solvents or ESI.
Yeah, so I just wrote on there, PDMS devices, they're made from Silgard 184, which is about one product you buy by a half liter. You mix it in a 1:10 ratio, and you put it in. If you want to accelerate curing, you put it in the oven for an hour at 70 degrees. If you don't have an oven, you just leave it overnight, and you've got a PDMS. So what you buy is the polymer base and a platinum-based initiator. And you just stir them together, put them over. There's nothing finished about it. And what you then only need to make a microchannel is something that's got a small ridge sitting on it. So if you've got a surface that's got a small ridge sitting on it, you can pour the PDMS on top. So you just pour it over as a big puddle, let it cure, peel it off, stick it to microscope light. And then there's I've done a lot in the area, but there's a lot of reviews that you can find in the literature about budget alternatives to microfabrication. So there's a whole range of options, and if you understand what you want to do, either etching or everything. As I said, if you don't care about the size, I've made devices in the past putting parafilm between two plates of plastic. So there are some really good ideas out there on how to make channels, and particularly if your application doesn't really require 10-20 micron. 100 things in the order of 100 micron are not that hard to make. And then it depends on what your solvent properties are, what the material is that you have to choose. But we'll be going through replication on, I think, Monday. Monday is our fabrication day, and I'll tell you how that's done in the clean room, but also how you can simplify life. It's my little box. So these ones are printed, were printed with a printer that's $400. So not still a lot of money if you need to pay it yourself, but it's not a $20,000-$30,000 instrument. This one here is immediately UV light as well, but that's made from a private film photo resist, using an office laminator to stick that to a bit of plastic. And this one is the same office laminator, but then first I have the electrodes being etched in the printed circuit board, which your department of electrical engineering is doing on a daily basis. This was a connectivity design, so we connected these to the electronics to apply an AC signal and record the reading of the other one. So this was a contactless connectivity.
And we have SDS page on chips as well. Like you said, that DNA can be separated with the microchips. We can separate protein as well, like SDS page we do in the lab.
Yeah.
There are chips and there are chips.
Yeah. So the actual bioanalyzer has got kits both for DNA and protein separation. It's both gel and electrophoresis. But I think this shops need to say a solution that
बस पानी कर दो
Sometimes in between 60 to 70 to